we believe that we will be able to pinpoint biomarkers for all major disorders, and I would start with AD in humans, that immediately can inform us about what kind of probably interventions on what kind of biomolecules we can start doing in human brain. As one of the ultimate goals in biological research is to have comprehensive maps of biomolecules in their environment, in tissue, with a high precision. So this field of technology is called spatial biology. And this is where I've been kind of working since I joined Westlake University, just because this is a very resourceful technology, and Westlake University allows me to explore and get these technologies in my lab. And it's all started from a simple idea that I've been exploring already over 10 years, when we're expanding the samples using a very simple concept, expansion microscopy. And it's been mostly done using microscopy, optical microscopes. We just look at the samples. And one day we came across a very simple idea. Can we apply expanded samples not only to microscopy but other analytical tools? And our first breakthrough in this direction happened when we, in collaboration with Gutianan lab, took a piece of expanded sample and were able to extract peptides that we were able to analyze with mass spectrometry. Expanding samples, getting it bigger, now allows you to improve a spatial resolution of analysis, right? If you imagine every time you get something 10 times bigger, it's way easier to go and pick a small part of its tissue. One fundamental goal of biological research is to map every single biomolecule in intact tissues, right? And there are many different things that you can do with it. But one particular thing that I'm excited a lot about is creating a molecular atlases of tissues. And I can compare this kind of technology to early age of the geographical discoveries in 15th century, right? When the people start traveling across Atlantic Ocean, like first travel happened from Portugal to India to find the way to actually deliver spices to Europe, right? And what was the critical point here? It was actually creating a maps so you can navigate from one point to another point, right? Maps being a major driver of this geographical discoveries starting 15th century. And I believe something similar will be happening in biological research when we have a maps of the tissues, and now we can start to navigate in the tissue that will help us to design and properly guide us through research process, right? It's way easier to get where you want to get if you have a proper map. So one of this application, and this is something that I hope my lab will be working in the next few years, is creating this detailed, comprehensive, and ground truth maps, or we can call it atlases of the tissues with a molecular precision and identification of not only proteome, but also other biomolecules, including metabolites, lipids, and transcripts. So why we need atlases? And again, this is exactly, so why we need atlases? So here's a very simple thing, right? When there is something going wrong in our body, right, we very often need to understand what biomolecules actually been dysregulated, what have the biomolecules been dysregulated, and where exactly they've been dysregulated. And very often when we look at under microscope on a tissue, we may not see the differences from morphological perspective. But creating this atlases with a molecular precision, we can immediately identify what biomolecules and where exactly, down to micron resolution, they've been dysregulated in our tissue, right? And once we know these biomolecules, and we know they are actually increasing in expression level or decrease, we immediately can use it for therapeutical approach for disease treatment, right? And here's another thing. I hope it's going to be also a very big step towards personalized medicine, where we now can develop actually this therapeutic approach based on particular patient data, right? We can take a piece of sample from a patient, understand what are these biomolecular profiles were, and immediately target that pathway, so these dysregulated molecules that actually happen in this particular patient, not associated generally with some of the symptoms, but actually too general, right? So this is probably one of the immediate applications here, and probably I'll give you one of the examples that we already apply this technology in research. I had a long interest in understanding the cause of Alzheimer disease in humans, but for ethical reasons we have to work with mice. And when we were brainstorming with Yilong about, oh, what we can use this technology for, right? What kind of new, this enabling discoveries we can make with it, I start thinking about very simple question. Can we identify that first protein, that first biomolecule in the cascade of molecular events that eventually lead to development of AD in mice? So we can kind of check the very first, very early event that goes wrong in mice, in the brain of a mice, that eventually leads to AD. And when we scan mouse brain, wild type, and as early as 2 months old transgenic animal that gonna have AD, we very quickly found one biomolecule that been dysregulated in a very specific area in corpus callosum that associated with synthesis of DHA. This is a lipid which is important for neuronal health, because it's one of a particular lipid that is crucial for neuronal membranes, right? This is, by the way, also DHA is one of the lipids that kids supposed to take in early age during very fast brain growth, right? And this is something that people haven't been discovering before, again, for very simple reason. There were no technologies that can allow, with such a high spatial precision, scan entire slices of mouse brain with a sensitivity to identify dysregulation of this protein. So this is one of the examples of how this technology can actually bring us closer to understanding on a molecular and cellular level causes of certain disease that can allow us now to actually target and maybe treat AD. So from my personal perspective, and this is where we are gearing up for our next step, we extending this technology to human samples, to those samples that actually collected biopsies, but now we can start looking at actual patient results, and we believe that we will be able to pinpoint biomarkers for all major disorders, and I would start with AD in the humans, that immediately can inform us about what kind of probably interventions on what kind of biomolecules we can start doing in human brain, right? Because right now we're trying to cure AD while we don't understand the mechanism of AD, right? It's kind of trying to fix some sort of engine or mechanism, but you don't know how to work. How does it work and why it's broken, right? Without understanding, it's going to be a random blind walk. But if we know what biomolecule being dysregulated and where exactly in the brain, it immediately makes our approach to AD treatment way more precise. And I believe that this technology will be a first step to identifying what biomolecules are dysregulated and where exactly in the brain they are located.